Yes. Can you hear me?
I can. You want your camera back on?
Sure. Except I'm not sure now how to do that.
You should be able to move your mouse down to the bottom of the screen and in the Zoom screen. There you
There
come.
we go.
There it is. Perfect. All right. Thank you everybody for joining us today at Hatfield's Research Seminar. My name is Cinnamon Moffett. I'm the Research Program Manager at Oregon State University's Hatfield Marine Science Center in Newport, Oregon, and I will be your host for today. A few logistics before we get started. You might have noticed that we have muted your mics and turned your cameras off and disabled your screen share. We ask that you keep it that way during the course of this event. We now have 46 participants joining us and the number keeps growing. So that just helps us kind of keep the noise on the screen down just a little bit. So we appreciate that. We do ask that you have any questions that you put them in the chat box. So if you move your mouse to the bottom of your Zoom screen, you should see a little chat pop-up box. You can put those in and we'll get to those questions at the very end of today's presentation. I'm sure Rachel is happy and willing to answer any of those questions. I also wanted to let folks know that we will be recording this talk today, and we will be posting it on our website. And I just put that information on where you can find the recording in the chat box, just as a practice if you want to see how to find chat. If you are taking this seminar for credit, Michael Banks is also on the line and will be helping monitor any questions. So ask away. Okay, so a few announcements and then for what we really are here for. I wanted to promote next week's seminar. We have Michael Barenfield, who is a professor of the Department of Biology and Plant Pathology at Oregon State University. And I'm really excited about his talk. He's going to be talking about building towards a new LIDAR era in satellite oceanography. And so right on the cutting edge of some really interesting things. And so I'm excited that we have him able to join us. If you are interested in any of our talks, we hold these talks every Thursday, same time, same place. But if you're curious about what talks are coming up, if you Google HMSC or whatever platform you use to find information, go to our homepage, log to the bottom, or scroll to the bottom, and you'll find our calendar of events. For today, though, we have Rachel Orban, who is the Seabird Oceanography Lab kind of head here at Hatfield Marine Science Center. She is a professor with the Department of Fisheries and Wildlife and affiliate faculty with Marine Mammal Institute at Oregon State University. She has her bachelor's in biological sciences at Cornell University and her PhD in ocean sciences from the University of California in Santa Cruz. Her focus, among many things, is on species habitat relationships and food web dynamics. And due to all sorts of COVID restrictions on travel, she has not been in the country for quite a while. So I'm very excited that we are able to bring her and have her share her latest research with us via Zoom today. So Rachel, I'm going to hand the floor to you. And thank you so much.
All right. Well, thanks, Cinnamon, for that introduction. And it's really nice to see lots of familiar names on my screen. So I'm really happy to be talking to you all today. I am, as Cinnamon mentioned, I am speaking to you from not from the Oregon coast, but from the sunny spring-like Falkland Islands. So hopefully my internet connection does this talk justice. So today I'll be talking about developing and employing biologging tags to sample the seabird's ocean environment. Oops. Just have to get the hang of it. So I thought I'd start out with a simple definition of what comprises the ocean environment. And personally, when I think about the ocean environment, the first thing that pops into my mind is the physical environment. So the temperature of the water column, the salinity, maybe some frontal structures on the surface, eddies, things like that. But of course, I'm a biologist. The biological ocean environment is also really important, especially if you're a seabird and you need to eat zooplankton or forage fish. And then finally, we have to throw humans into the mix. So there is an anthropogenic ocean environment. We've hopefully not just begun the industrialization of our oceans, but we have done a good job of developing global fisheries, and then also in our nearshore waters, offshore oil platforms, wind energy development, things like that. So a foraging seabird no longer goes out to sea and encounters just non-human entities. Now they might encounter fishing vessels and other things. So my talk is about using biologging as a useful tool to sample this ocean environment. And why would we even want to do this? And one thing that I think is important to keep in mind is that marine predators can act as these sentinel species. So a seabird depends on forage fish or zooplankton that are then dependent on phytoplankton and associated environmental conditions. And as an ecologist, when we look at seabirds, we can follow their reproductive success, phenology, things like that, and better understand the ecosystem. When we place biologging tags on birds, we're then tracking them to places that are biologically relevant for them. And say a diving seabird would subsequently sample these biologically relevant places. But it is also important to consider what we're doing when we tag animals. So it's really been in the last, I guess, 20 or so years where biologging has become general practice. It's really opened our world in terms, especially as seabird ecologists, without biologging, it's not possible to really know where individuals are going. And we know that seabirds are making incredible migrations, pole to pole, things like that. So understanding these individual movements is really important, but it's also important to consider what we can do to improve the design of the biologging tags that we're using. And we've learned a lot in terms of knowing what weight or what shape, how to attach tags, these things that we can improve and then really tailor them for different species characteristics. And it's also important to consider how a tag is actually sampling the data and how that might be specially adapted to the animal that it's being used on. So I've included a couple of figures from a meta-analysis of looking at the impacts of biologging tags on birds. So you can see that the movement modality does influence the effect size in terms of both survival and reproductive effects. It's different between birds that soar, are flightless penguins, and birds that are flapping or flapping and soaring species. So your general biologging device has some really important components. We need to consider the tag size and shape, how it's attached to our animal, and how it's powered. So recently, tag developers have been able to add solar panels to some tags, which increases their longevity. It's also important to know how the data is being transmitted. So tags can be archival, so you might need to recatch an individual animal to get the data back, which in some situations, so for seabirds, in particular albatrosses, they're really site faithful and they're very easy to catch. So having an archival tag has some benefits. But in other cases, you really need a way to transmit that data off the tag in the volumes that you're interested in. And along with that, it's really important to have flexible programming, especially if you want to add multiple sensors to tags. So typically, we've been using biologging tags to better understand animal space use, behavior, movements, and this has improved our ecological understanding, especially in the context of seabirds. And we can use the spatial data in particular for things like marine spatial planning. But we can also, with the right design of biologging devices and also associated data processing, we can start to measure in situ environmental conditions. So the conditions, the physical, biological, and human conditions of the ocean that the animal might be encountering. And this has some benefits in terms of our understanding of an animal's ecology, because we can actually measure these things at a resolution that is relevant for that animal as it's moving across the ocean. But at the same time, these data streams can actually be useful for other purposes. So, for instance, an animal might be collecting water temperature measurements that could be used in climate oceanographic models. We could use these data to ground truth measurements from satellites. Later in this talk, I will discuss an example where we're actually where tags have been developed to help enforce human activities. And I think this is just the beginning of how biologging data may be used in the future in sort of situations that we haven't even thought of yet. So today I'm going to talk about two current projects that we have. The first one is the Cormorant Oceanography Project, and this is really a project that is designing tags fit for purpose for cormorants to better measure their physical environment. And then the second project I'll be briefly mentioning is one where we're using biologging tags that detect the radar signals from vessels that albatrosses encounter. Now I wanted to mention, I just had a new graduate student join the lab, Susie Winquist, and she will be doing her master's in the Fisheries and Wildlife Department on the Adelie foraging behavior using video biologging tags. So the penguins that she's studying will be taking her and us, hopefully, on a tour of their underwater world. And this is just another way that we can use biologging to better understand the ocean. So in a few years from now, I'll have a third study to talk about. So using biologging tags to measure physical ocean conditions is not a new idea. It's something that's been around for probably over 2 decades. But the workhorse tags for marine mammals in particular, I have them here. So we have splash tags made by wildlife computers that carries a fast response temperature sensor. And then the CTD tag made by the Sea Mammal Research Institute or unit at the University of St. Andrews. And this tag has both a fast response temperature sensor and conductivity. And these tags were deployed first roughly in about 2003, maybe a little earlier, I'm not 100% sure, but they've been around for a while, which is great. And they're using data from elephant seals are being pushed into models and things like that. But the tags themselves are big. So at 400 grams, that's about the size of a puffin. So that really doesn't give Seabird researchers the tools that they might want to have if they want to try to use this biologging environmental sensing approach. But there are some Seabird examples. So one of the oldest papers that I found was published in 2000, where they used temperature data from foraging king penguins from Kerguelen to map the polar front. And there's been a few other, mostly penguin-based studies that have derived temperature profiles along for dive profiles. Seabird researchers have also noticed a few other things in their biologging data. So there's been a couple studies now where they've realized that seabirds do rest on the surface. So birds like razorbills, murres, shearwaters will sit on the surface of the water and can drift with the surface currents. In some cases, especially there's a shearwater example, they they were able to disentangle how much of that drift was from wind versus surface currents. The razor bill example, which is the plot at the bottom, Cooper et al. 2018, they seem to do a bit better job of mapping surface currents, and it could be because they are a diving bird. They're coming to the surface and resting at the surface, versus a shearwater might be a little bit more buoyant and active on the surface. And then the last example I think is quite amazing, but researchers have been able to use the stereotypical flight patterns of shearwaters and albatrosses and then disentangle both wind speed and wind direction from these flight paths, which is pretty amazing. And we're not doing that with cormorants. They don't fly in that dynamic soaring style. So for our cormorant oceanography project, I'm the Seabird researcher side of the project, but we're collaborating with 2 lab groups in CIOS, both physical oceanographers and then also a group that is using the data to assimilate our in situ data into their models. So our goal is to develop tags into a low-cost, long-term way of measuring these oceanographic variables. And we're trying to do this in a logger that is at least less than 40 grams. Ideally, it might be about 20 or 30 grams, still too large to put on something like a common murre, but we're getting closer. And ideally, these tags would be able to send us data in near real time, and then that data can be processed and accessed very quickly after that. And then, of course, trying to get these data to actually be useful or in a form that's useful for ocean modeling. Why cormorants? One of the reasons is that they are a generally large-bodied diving bird, so they tend to be over 100 or 1,000 grams. So they are able to carry larger tags, which is helpful, especially when you're starting out. It would be very challenging to start this project focusing on a species like the common murre, where maybe a tag that's 5 grams would be more appropriate. There are a number of cormorant species and shag species that are found globally, and they're found on all of the continents. So there's options in many of the nearshore waters. So these birds, like the Brandt's cormorants found off the Oregon coast, are using really the nearshore habitat, and they're using areas that are very dynamic and complex. So with river mouths, estuaries, places like that. And the other benefit with cormorants is that they make a lot of dives. So we found that our tag birds so far are making about 100 dives per day. So that's a lot of temperature or conductivity profiles. So our current version of the tags are made by a company based in Lithuania called Ornitella. And we've been able to work with them to add both a conductivity and a external temperature sensor. And both of these are still in progress, but we had some good results from a deployment we did this summer in the Gulf of Alaska. So the tags themselves are transmitting data to the cell phone networks. They have solar panels for recharging, GPS for location fixes, pressure sensors, and they also carry 3-axis accelerometer and magnetometer. And they have very flexible programming, and they can be reprogrammed on the fly. So we can access the data through an portal on the web and reprogram the tags whenever we wish. And the tags are calling in about once a day. And we can adjust how frequently they try to communicate. So what variables are we trying to derive from the movement patterns of this flying diving bird. It's because they are diving birds, they do sit on the surface between their dives. So this allows us to use GPS fixes, consecutive GPS fixes to measure surface currents. We're also pretty sure that we'll be able to measure wave statistics using the accelerometers. There's some variety in how much cormorants forage benthically, but for instance, the Brandt's cormorants do a lot of benthic foraging, so they're going to the bottom, and then their dives essentially are a depth sounding. So we're using the shape of the dive, so a flat bottom dive, to indicate a bottom dive, and then we're able to georeference that as soon as the bird comes back to the surface and takes a GPS fix. I mentioned temperature profiles, so getting an external fast response temperature sensor on the tags, and then that should also allow measurements of air-sea temperature contrast when birds both dive down into the water and pop back up to the surface. And finally, we are very much hopeful that we can get a working conductivity sensor on these tags. Excuse me. So this is just a schematic, basically of just what I went through, but it is important to note that cormorants are very coastal, and this does allow for transmission of large amounts of data to the cell phone networks, and this can happen daily. So this is really important, especially since we're collecting temperature and pressure at 1 second intervals. We'd like to collect accelerometry data at about 5 hertz. So that amount of data can really stack up. So the next couple slides are some of the data processing that we've done so far in terms of these different parameters. So this is a slide just demonstrating a transect. You can see on the top plot, this is the mouth of the Columbia River, and there's the red dots are the dives that are shown in the bottom figure, and the USGS bathymetry is the white line. So you can see the birds are tracking this pretty well, but they'll occasionally dive below that line. So the good thing about the birds is that they will dive to the bottom, but they're not going to dive through the bottom. So it's unlikely that we would estimate that the water column is deeper than it actually is. We certainly, depending on how good or how stereotypical bird dive shapes are, we could estimate that the bottom would be higher than it actually is. So we've done some preliminary work with using the accelerometers to decompose wave statistics. And this was done with a duck decoy wearing a tag in the Hinsdale Wave Research Lab. And we were able to use the accelerometer to measure the waves that the duck was experiencing. And we think that this should work on our free-ranging cormorants as well. But it is certainly a challenge when you don't know exactly what types of waves the birds are encountering to sort of ground truth this. The next measurement are surface velocity estimates, so surface currents. So for these, we're really at a stage where we're trying to design the tags to better sample these measurements. So some of this data came from archival tags that were sampling at a higher GPS fix rate, but we can't have our tags sampling at 30-second intervals all the time. That's just too much power consumption. So instead, we're trying to link the GPS fixes and really target them for when birds are resurfacing. So the tag the firmware for the tag is being redesigned so that when a bird resurfaces, this surfacing event triggers the GPS to do what the tag manufacturer calls a burst fix. So we'll get a series of GPS fixes within a 10 or 15-second window that we can then use to estimate these surface currents. And that will be a really helpful way of targeting when birds are sitting on the water. So cormorants tend to have a wettable plumage, so you see them around drying their wings a lot, so they're not spending large amounts of time just sitting, resting on the surface like some other species. So albatrosses will sit on the surface for most of the night resting, but cormorants don't really do that. So they're really on the water for much briefer periods of time, and we can use that to our advantage to target our sampling during those times. So I mentioned we were able to do some field work this summer. We were very fortunate to be able to send a team of 3 people to Middleton Island, which is located in the Gulf of Alaska. And it's a really interesting place to work on seabirds. It's a almost laboratory-like setup where the birds are nesting on the outsides of these towers that you can see in the bottom of the photo. And it's very easy to access nesting birds. Each nest has a little door that you can open and you can actually catch the bird just like that. So it was very easy for us, the crew, to catch pelagic cormorants, deploy the tags, and then a couple of weeks later recatch birds. And this was very helpful for us, especially as we're testing the fast response temperature sensor and conductivity sensor to be able to have the tags again and take them back to the lab and see what's going on. And this is an example of the temperature profiles that we were getting off the tags. So for our next steps, I think I've alluded to this a few times, but we're planning on continuing to refine the tag programming to really target the data sampling for when we want it. And we also need to modify the temperature and conductivity sensors to improve them. We're currently working on developing an automated processing of the tagging data in collaboration with the Animal Telemetry Network. And we are planning and hoping to deploy tags on new species in the next year or so, COVID willing. So that's where this project currently is. And I think, yeah, I'm really excited to see what happens over the next year. We've certainly made a lot of improvements with the tags in this last year. So I'm going to switch gears and talk about the second Biologging project. So this project is just getting started, and similar to the Cormorant project, it also has a pretty strong and important field component that we're still not quite sure of how likely it is to happen, but the general goal is to be in the field in February, working with albatrosses in the northwestern Hawaiian Islands. So the project is to really better understand the occurrence of fishing vessels within the Papahana Mokuakea Marine National Monument in the northwestern Hawaiian Islands, and also to understand when, why birds are encountering, or not encountering, but approaching fishing vessels, because approaching a fishing vessel makes them more vulnerable to bycatch. So fisheries bycatch is a significant conservation issue for marine predators, particularly seabirds, and particularly albatrosses. And this holds true for the species in the northwestern Hawaiian Islands. Certainly the bycatch rates of black-footed albatrosses have been increasing in recent years. So generally, especially from the biologging perspective. I have all these tracks of where animals, seabirds are going, and I can easily relate these tracks to environmental conditions measured from satellites. So not quite the fine scale data that we're getting from the Cormorant project, but still I can have a sense of what that type of environment is like for a seabird. But I don't know when birds are encountering boats, or at least I didn't until the advent of Global Fishing Watch. So Global Fishing Watch, if you haven't explored their data set, you really should go online and check it out. And it's a data set that has, declared vessels. So vessels that are carrying AIS transmitters, and then they're able to basically use movement algorithms to identify fishing vessels and actively fishing vessels to better understand global fishing effort. But there's still some holes in this data set. So small vessels might not carry AIS, and certainly illegal fishing, those vessels probably don't have AIS transmitters operating. So for this project, we're planning on using very special biologging tags that were designed to detect radar signals from vessels. So when a bird approaches a vessel, the vessel is probably transmitting navigational radar, and this will trigger the tag to change its sampling interval and also denote that it has in fact detected radar. And we did a trial deployment of this type of tag a few years, I guess in 2018 at Midway Atoll in the Northwestern Hawaiian Islands, and we're able to detect a couple fishing vessels that birds encountered. We also learned that the airport on the island turns on radar when the planes are coming, which is about once every 2 weeks. But these tags were really developed by the French to be used in an operational format. So the vision is to use wandering albatrosses to patrol the waters around their breeding colonies in the Indian Ocean. And these tags, there's a couple different versions of the tags, but this one, the operational version of the tag will transmit a radar detection to the Argos satellites that then gets processed and cross-checked with the locations of birds carrying or boats carrying AIS. And then if it's an unknown vessel, the French can send a Navy ship or a fisheries patrol vessel to go and check out what's happening. We're not planning on doing this in the Northwestern Hawaiian Islands, but we are planning to see how this type of bird surveillance what it might look like for that area. So the two species that nest in the Northwestern Hawaiian Islands are the Laysan albatross and the black-footed albatross. And I did mention that the bycatch of black-footed albatrosses is increasing in both the Hawaiian and Alaskan fleets. And generally, the impact of high sea fisheries on both populations is unknown. And the goal of our project is to provide information on when and where these birds are encountering vessels. We'll cross-check these encounters with the Global Fishing Watch dataset so we can understand what type of vessel they're encountering. And then we can use this data to better understand the drivers of albatross fisheries interactions. And this work really fits nicely off of a project that Lee Torres and I are just finishing up, where we used regular old GPS data and overlaid it with the Global Fishing Watch data set to do just this. But in particular, we had lots of tracks for black-footed albatrosses, but it was quite disappointing that we only had a handful of encounters. So we really weren't able to disentangle the drivers of why this species in particular may approach boats. We just were able to see that it approaches boats more frequently than the other two species. So one of the motivations for this next study is really to increase the sample sizes of this data set. So I did mention that part of this project is to use albatrosses to see if there is illegal fishing within the monument waters, and then also just generally patrol the adjacent high sea regions. And often the albatrosses aren't actually foraging within the monument, they're transiting across, but they do, from their breeding colonies, they cover a lot of that area just with those transit flights. And then we'll be able to use this data to hopefully better understand these albatross vessel interactions. Finally, this slide sort of lays out the plan for the project and how we're planning on combining the biologging data that better describes these encounters with vessels. So without the vessel data, we really don't know if birds are aggregating in places because the food is naturally abundant, or if they're there because there's fishing vessels. And we certainly see, so in the Global Fishing Watch data, you can see the border of the monument highlighted just in the fishing effort. I think this is about 6 months of effort. So there is that, there could be a border effect in terms of how vessels are concentrated and what albatrosses might see when they head out to sea. So we'll be able to combine all these things together and really get a better sense of the human environment that these birds are encountering. So that's, just checking my time, but that sort of concludes my talk. I mentioned at the beginning of this section that we're hoping to do some field work on the Northwestern Hawaiian Islands in this coming February when the chicks are just newly hatched, but that's still something that we're thinking about. So with that, I think I will take any questions.
Thank you so much, Rachel. So we definitely have some time for. Sorry about that, I muted myself. I was just gonna say we have some time for questions, so please feel free to put them in the chat. As people are typing things in, I had a quick question for you, Rachel. And going back to your cormorant study, can you give me kind of an idea of, if you were looking at biological behavior, how many tags would you have to have on your cormorants to really understand the difference between individual behavior and community behavior?
Cormorants are interesting. So there's a couple levels, I guess I can answer that at. So I just, from sort of the tagging perspective, or like the individual movement perspective, you know, if you have a population of birds that all do different things when they're foraging, then you would need more tags to sort of characterize the foraging area of that colony. So if everybody does the same thing, you only need to tag a couple birds, you know what everybody's doing. If everybody does different things, you need to tag more animals. And then you can also sort of take that apart where, you know, you have males and females, so they might be doing different things. So maybe you'd want a decent sample size of both. But cormorants do different species do some group foraging. So you could get, you know, if you tag enough birds, you could get individuals that are actually foraging together. So we tagged Brandt's cormorants at the mouth of the Columbia River in 2019, and we tagged about 20 individuals. And within the Columbia River, everybody was pretty much using the same areas around East Sand Island and out towards the mouth a little bit. But what was really notable and what we certainly didn't characterize was their longer distance movements. So we had birds moving from the Columbia River around the outer coast of Washington. We had a few birds go sort of back around. My geography, all my place names are a bit rusty, but back around into the Salish Sea and south into the inland waters of Washington, up past Victoria. We had one bird hung out at the ferry terminal where you would catch the ferry to go to Vancouver Island. And then another bird went farther north. And then we had one bird that decided to go south and went all the way south to Long Beach, California. So by no means did we really characterize those types of movements with our sample size.
Nice. Okay. We're definitely getting some questions, so I'll start to get through them here. Michael asked, can you talk a little bit more about solar powered tags, the constraints or advantages of that type of tag?
I think the one sort of big constraint is that you do need to be able to place the tag on your animal in a way that the solar panel is exposed to the sun. So with a bird that has feathers, it's a little bit challenging in terms of making sure that those feathers don't obscure the solar panel. So for instance, we did use tags very similar to these that we used on the Cormorants on Western gulls, and we chose to use a leg loop harness system, which places the tag a little bit farther down on the back of the bird, but then the wings do cover the tag when the bird is standing around. And it turns out, especially in Oregon, Western gulls do a lot of standing around. So when they have chicks, they're quite busy and they make decent, beautiful foraging trips, and the tags charge up really well. But then during the winter, the birds just stand around and the tags, the battery voltage drops out. So that's one of the biggest challenges is just being able to make sure that the panel is exposed. We did find that the pelagic cormorants are a little bit harder on the tags than the Brandt's cormorants. So some of our tags from them came back with pretty big dents in the solar panel. Like they're covered, I think, with like an epoxy thing. So we don't think that the solar panels themselves were damaged per se, but there is that chance. Maybe eventually it would happen. But the biggest, I think, advantage is that you can really get much smaller tags. You can put much smaller batteries in tags. And then, if your tag is staying on for a long time, the tags that we're using do regulate their sampling intervals based on the available power in the tag. So when it's sunny out, the tags are chirping away. They're taking lots of GPS fixes. But when it's not, the tags can drop down to one fix every half hour or one fix an hour and conserve power that way and still give us the data that we want.
So I think that leads us to this next question quite nicely. Jasmine's asking just this kind of technology, how do you deal with outliers and being able to determine if it's a natural behavior or a true outlier? And you were just talking about variation in data sampling. So how do you deal with your data and the variability in that data? And that might be a big question.
Yeah, I guess I find the outliers always quite interesting. So it is, I think it is a tendency of us, the bird that went to California. Like, I think that's such a cool story. And I don't know, maybe he's the only one. I really don't know. So it is challenging, in terms of if you want to characterize, say, the habitat or like the core habitat area of a population. But we also really do struggle with getting even sample sizes from like the whole annual cycle or multiple colonies. It's really challenging. Like if colony, for seabirds, like if nearby colonies will have sort of discrete foraging areas. So you're going to struggle in general to encapsulate everything and all the variability. So I think, those outliers sort of speak to the unknown, and we just don't know what everybody's doing yet. And I guess there are some ways to look at the data and like the movement patterns to see, or at least try to understand or extrapolate from the data if the instrument itself is having an effect. So we can look at like daily distance traveled, or make sure that the birds are still diving. Like that's quite important for cormorants. If they're diving, that means they're eating. So we can look at things like that and see generally how they're doing, and if they're outliers in that way or not.
Nice. So this question goes specifically to your albatross study. Since the tags detect a radar signal, can they be modified to also detect the AIS, so that you wouldn't have to depend on an overlay?
Yeah, so that's, I think, the next step, and so the tags are being designed, like his last name's escaping me, but Dominique, who's based in New Zealand, Sextant Technologies, and I think his next design of the tags is to detect the radar and the AIS transmissions from the vessels when the birds encounter them. But it is really, in terms of those tags, like I didn't really address it in the information I gave you, but they're transmitting data to the Argos satellites. So they're really limited on the amount of information they can transmit with the power that they're able to carry on the birds. So it is a bit of a balance in terms of size of the tag and what information they can collect. So with the albatrosses in the northwestern Hawaiian Islands, they're a smaller bodied species of albatrosses, so we need smaller tags. So I think the AIS detectors will work for the wander albatrosses first and not be small enough for our use.
So that leads actually to the next two questions. They're both talking a little bit about the impact of the tags on the birds themselves. So have you noticed the tags altering the bird's behavior, or why certain species might be more sensitive to tags? Can you speak a little bit to that?
I think generally albatrosses are able to handle tags quite well, especially we're aiming for tags well, at least below 3% of their body weight, but preferably even smaller than that. And for the albatross, we'll be attaching the tags with Tessa tape. So their tags will be taped to their feathers, and then they'll lose them when they molt their feathers. Or in some cases, we will be using archival tags. So the tag will be on there for a foraging trip or two, and then we'll remove it when the bird comes back to feed its chicks. The cormorants, it is a bit more challenging. They're hard to recatch. So there's a lot of unknowns in terms of long-term effects of carrying tags for that amount of time. So we're attaching the cormorant tags with a wing harness, and generally from what we've been able to see, so we haven't noticed any adverse effects of that harness system, but I mean, things happen. So there's definitely, I think, that chance of something detrimental happening to the bird when we're asking it to carry that system. But it is something that I think we're continually trying to refine the tags that we're placing on the birds, and also the attachment method that we're using. And that was one really good thing about working at Middleton Island with the pelagic cormorants, is that we were able to recatch them there, which is a pretty special situation.
So these next two questions are both kind of related to how the tags themselves work. This particular question is wondering, for near surface measurements, how soon after the bird enters the water is the tag starting to record, or is it always recording? So can you talk a little bit about the limitations of the near surface data from the sensor and the delay on when it starts to record?
Yeah, I don't quite remember what the pressure threshold is. I think it's like 2 meters or something around there, but I'm not 100% sure. And I'm not 100% sure, how that's exactly being triggered. If it is, I think like the pressure sensor in terms of power management doesn't actually consume that much energy, so it could be just checking all the time and then just logging it when it goes underwater, which is good for the data transmission. It really improves that side of the coin. We don't need pressure sensor data all the time, but the tags are able to basically every time the birds dive, they are recording the pressure and then the associated temperature and salinity data. And there is a bit of a delay when the birds resurface for the GPS to refine satellites and then take that fix.
So you're doing a good job of leading from one question to the next. So how do you deal with, like on the Cormorant study, when you were dependent on cell phone reception, how do you deal with areas if the birds are in a place where cell phone reception is limited?
The tags are able to store quite a large amount of data on them, so they are able to go for a few days, weeks without actually transmitting data, and they'll just archive that data and then send it when they can. So we're sort of trialing that now. We have a handful of pelagic cormorants that we didn't recapture and still are carrying tags, and they all went to Southeast Alaska and northern BC. So there's one foraging off the north shore of Haida at the moment, one I think in towards Prince Rupert, I think, and I think another one sort of the inner waters, definitely not all the way to Juneau, but just on the inside passage side of things. And cell phone reception is there because I know where these birds are, but it's not great. So the tags do send us SMS messages with a most recent fix, so we know that about where they are, but it's not sending us a lot of data. And then when they do have better cell phone reception, they will send more. So it works pretty well.
Nice. So this question jumps back to your albatross. Can you talk a little bit more about the management options that you perceive if that project is working in the way that you think it could or might?
Yeah, I sort of glossed over that because I was framing the talk around the biologging side of things. But I think in general, a lot of how we've approached bycatch and bycatch mitigation is from the perspective of the fishing vessel and what we observe from the vessel. So I'm not saying that the bird side of things is going to be the golden bullet that solves all our problems, but it does seem like it's a good place to look for some innovative solutions, or at least a different perspective on the same project or problem to see if we can improve how we manage. So understanding when, why, where birds are approaching fishing vessels, I think could give us the tools to predict when, why, where birds are, or when and where birds are approaching fishing vessels. And if we can predict where bycatch events might be more of a problem, then we could potentially come up with some ways to alert fishermen in those regions, things like that.
Nice. You're getting a lot of just great presentation. Thank you so much for sharing. I think we've covered everybody's questions that are in the chat currently. If there's any last little questions, go ahead and put them into the chat. Otherwise, Rachel, I just want to thank you so much for making the effort to join us from afar. It's one of those little silver linings of Zoom. It just allows us to do things that we never thought possible until we were forced to think about it. So I sure appreciate you joining us. And for those folks that are online, thank you for being with us. Feel free to join us next week, same time, same place. And I think, Rachel, you are open to folks contacting you directly if they have specific questions about your project?
Yes,
okay.
Of course.
And so if you are unable to find her contact information, go ahead and reach out to me and I'll make sure to share that. Unless you want to put that in the chat real quick, Rachel. I don't know if you can see chat in your format right now. Yeah, okay. So for those
really know how.
reach out to me specifically and I will help you connect with Rachel. And again, thank you everyone so much. And Rachel, thank you for joining us. I appreciate it.
Yeah, thank you all for coming. It's great to, it's great to be closer to everybody in Oregon, even from so far away. So.
You're getting claps and now high fives. So I don't know how to do that virtually, but that's what we're getting in the chat. All right, everybody, I'm going to go ahead and close this down. Thank you so much for joining us. We'll see you next week. And thank you, Rachel. Bye now.